everyone, and welcome to the Paper Trail. My name is Bria Macklin, and I'm a postdoc at the Gladstone Institute. The Paper Trail is an online interactive journal club that was developed from 4 former colleagues turned lifelong friends: Dr. Quentin Smith, an assistant professor at UC Irvine; Dr. Michael Blatchley, a postdoctoral fellow at CU Boulder; and Franklin Hall, a graduate student at Johns Hopkins University. Born from our love of discussing science and our yearly paper a day challenge, we came up with the idea for the Paper Trail to be a platform that allows primary authors to come and share the story of their research. And with that, I would like to introduce our speaker, Dr. Leah Elias. Leah received her Bachelor's of Arts in Neuroscience and Behavior from Mount Holyoke College. She completed her thesis work in the lab of Dr. Ismail Abdus-Sabor, addressing the peripheral and central encoding of pleasurable social touch in mice. She currently works as a postdoc in the laboratory of Dr. Seth Blackshaw at Johns Hopkins University, using single-cell transcriptomic approaches to understand mechanisms of homeostatic sleep pressure. And so with that, I'll let Leah tell us about her work.
Yeah, so as Dr. Macklin said, my name is Leah Elias. I'm currently a postdoc with Seth Blackshaw at Johns Hopkins, but today I'm going to be talking about my thesis work that I did with Dr. Ismail Abdus-Sabor. And so, yeah, this is a paper that has come out recently, and it's entitled Touch Neurons Underlying Dopaminergic Pleasurable Touch and Sexual Receptivity. So before I get into all the science, I need to highlight a few of the really key players of this work. So without whom this really wouldn't have been possible. So Isabella Succi worked alongside me for several years on this project, first as a summer undergrad from St. Joseph's University, and then as an Abdus-Sabor lab technician. She's now a PhD student at Columbia. So Bella conducted, along with me, a lot of the behavioral experiments, in addition to, and including the fiber photometry. And importantly, when the lab moved to Columbia, she really led the revisions effort and got all the experimental techniques set up at Columbia, and helped everybody play the role that they played.
I imagine that was very stressful, like moving your mouse colonies, this whole entire setup to an entire university, and then you not being there.
Yes,
yeah.
There will be a whole section at the end about
great.
The stressors and the uncertainties of the PhD, and how, you know, it works out, but yeah, challenging. And I think Bella was really integral for making that possible. And yeah. Melanie Schaffler was a PhD student along with me in the lab at Penn in the neuroscience program. She's now working for Teva Pharmaceuticals. Melanie conducted the condition place preference assay, which is a behavioral experiment that we'll talk about. And in her own thesis work, she found some really interesting complementary findings about these same social touch neurons. And that work is not yet published, but should be coming out soon. So really, really critical role there. William Foster was a technician in the Abdus-Sabor lab, and now is a PhD student at Columbia. And Will found the kind of initial finding alongside me that sparked this paper, really inspired it. And then periodically assisted with behavioral experiments, and also taught us how to do the ovaryectomy surgery, which we'll get into as well today. And of course, Ismail Abdus-Sabor, my PhD mentor and the principal investigator of the lab and corresponding author on the paper. We couldn't have done it without his leadership. Yeah, so I'm really grateful to have worked with this team, not only because they're brilliant scientists, but they're also phenomenal people. And it's really special when you can call your colleagues your friends. So we can really thank Ismail for hiring good people that really made this work possible. Of course, there's so many other people that contributed to this work. Really importantly, Dr. Victoria Abrera's lab at Rutgers, really critical collaborators. Mark Radwell, a postdoc in the lab, conducted the vast majority of electrophysiology in this work. Manoba Heek did the viral tracing, as well as some of the immunostaining and in situs. Akira Fushiki, a postdoc from Rui Costa's lab, did the Inscopics experiments. And then just so many other people in the Abdu Sabor lab, each playing a role.
And so I'm wondering if you could tell us a little bit more about how this team came together. Was it a thing where maybe you were just initially working on this project, or it was already a super collaborative effort that you kind of joined in on? Or did you guys kind of put the pieces together as you realized what you needed?
Yeah, more so the last thing that you said. It was, yeah, like I said, so Will and I kind of during my rotation in the lab found the initial discovery, and I'll talk about this in the timeline. But yeah, we found this initial discovery that the mice will lower their backs in response to the activation of these neurons, and that just was like, what's going on? And so as we realized that something interesting was happening, we kind of pulled on more and more people to the project. And yeah, once we had that initial discovery, this kind of really became my central thesis project. And then others in the lab, we're all part of the team. And especially for the revisions experiments, once I was 2 states away and everything was spread out across the East Coast, we took on the collaborators, and all the other people in the lab became really, really helpful for getting this done.
Yeah, having a good team is like crucial when you get to that revision step.
Yes.
And I have to say, like, when I think of a scientist, when I went into grad school initially, this is not the scientific team that I thought of. I mean, this is beautiful. I think it's just amazing to see the group of people that came together to just get this amazing finding. And I think just this slide here is so inspirational. Like, I think this is amazing. So awesome.
Yep. I had the best team. Yeah, I couldn't have had it any other way. Okay, so I think I'll move on into the science. So kind of the big picture question that underlied this paper was, how does the skin detect socially relevant tactile cues? So when we embrace a loved one, how does that simple pressure tell the brain that this is a meaningful, positive social interaction? The why a hug feels good is a really interesting question in itself, but it's also become increasingly critical as we emerge from a pandemic, where we're beginning to see the devastating impact of social isolation on mental health. But the impact of social isolation or dysfunctional social touch on mental health and well-being has been an interesting question long before 2020. So for decades, we've known that, for example, skin-to-skin contact between newborns and a mother is critical for the healthy cognitive and behavioral development of the child. And we can also think about situations where social touch isn't functioning in a normal and healthy way. So many individuals with autism spectrum disorder will find that social touch is not pleasant. And similarly, we can also think about various forms of female sexual dysfunction in which, broadly speaking, intimate touch is not sending the appropriate signals to the brain. So social touch is really critical across development and for various forms of social relationships. And so it's really important that we understand how healthy social touch gets from the skin to the brain in order to identify therapeutic targets for what happens when it's missing or when it's not functioning properly. Generally speaking, we know that social touch gets from the skin to the brain via the somatosensory system. So mechanical stimulus, like a touch to the skin, will activate a sensory neuron, a touch sensory neuron, which has its cell body in the dorsal root ganglia outside the spinal cord. So we call these ZRG neurons. And so it innervates the skin, and its job is to transduce that mechanical stimulus, like a deflection of the skin, into an electrical stimulus that then travels down the axon to the spinal cord. This sensory neuron then activates a projection neuron in the spinal cord that sends the electrical signal to the brain, where it can be interpreted as pleasant or warm or soft.
So is that one neuron? So does one neuron kind of travel that length from, let's say, your hand to the spinal cord, or is it like multiple ones?
Great question. So it can be a bundle of neurons, but it is one synaptic connection. Yeah. So these are like the largest, like longest, cells in your body, from the end of your pinky toe all the way to your spinal cord. So it can be like a meter long.
Wow.
It's remarkable. Yeah. Yeah. So this is an oversimplification, but if we look at even just the level of the skin, there's just a huge variety of these sensory neurons that are each specialized for different types of touch. So from pain to temperature to motion to texture. And so our question then is, well, is there one subtype that might be specialized for social touch? And so in humans, we know that we think there are, and they're called C-tactile afferents. And I'm going to kind of tell you 3 things about these dorsal root ganglion sensory neurons, C-tactile afferents. So firstly, as their name suggests, they are C fibers. So of the different dorsal root ganglion neurons, the C fibers have the smallest axon diameter, which means that they conduct slow signals. So C-tactile afferents are a subset of C fibers. Secondly, C-tactile afferents innervate exclusively the hairy skin, so not the glabrous skin like the palms of your hands or the soles of your feet, but the hairy skin with these large arborizations that are thought to optimize them to detect motion across the skin, like a socially relevant stroking stimulus. And thirdly, C-tactile afferents are thought to detect the pleasantness of social touch. So rather than the discriminative touch, like this feels rough or this feels smooth, they detect the more affective component of touch, like that feels pleasant. And so that comes from studies where they find that the stroking velocity that optimally activates C-tactile afferents is highly correlated with the stroking velocity that participants will rate as pleasant. And that's like 3 to 10 centimeters per second, if you're curious. And so taken together, we can call these C-tactile afferents these pleasant social touch detectors. But to really understand how they're sending signals from the skin to the brain and all the mechanisms there, we need to be able to experimentally manipulate these neurons. And so for that, we have turned to the genetically tractable and highly social mouse model. So in the mice, we can identify molecularly distinct subsets of dorsal root ganglion neurons, which allows us to make specific manipulations in specific cell types. And then we can observe how the behavior changes. And so for this paper, we're really interested in the dorsal root ganglion neurons that are molecularly defined, that express this particular protein called, I'm not switching my slides. Oops. Yeah, okay, called MRGPRB4. And so I'm going to call these the B4 lineage neurons for short. So I'm interested in these B4 lineage neurons because they share a lot of similarities with human C-tactile afferents. So on an anatomical level, they are also C fibers, and they innervate the hairy skin with large arborizations. There's also evidence that they respond to stroking stimuli. And there's evidence of a conditioned place preference. So this means that mice will prefer to spend time in a chamber where these neurons were previously activated. And so that suggests that their activation is pleasant in some way.
Can I ask a question really quick?
Yeah.
So you mentioned in this slide and the previous slide this stroking stimuli, but I'm wondering if there are any other types of touch that these neurons are able to perceive as pleasant.
Ooh, yeah, that's a good question. Yeah. So this study identified that, yeah, a paintbrush stimulus would activate these neurons, but like a pinching stimulus with forceps would not. And yeah, in our work, we didn't test too many mechanical stimuli to the skin to look at changes in the activity of these neurons in particular. It's a technically challenging experiment to do. But one of the other kind of gentle touch stimuli that people will try to do is air puffs. So it's different than a stroking stimulus, but the air puff is an interesting mechanical stimulus because what it does is it deflects the hair without necessarily deflecting the skin. So you can assess whether your sensory neurons are detecting just the movement of hair deflection or if they're detecting deflection of the skin. And so we did test the air puff stimulus when we got rid of these neurons, and I don't think we saw a major change, but that could be because there's other neurons that are compensating that also detect air puff.
So sorry, I apologize for my naivete with this. So there are, I guess, sort of different types of neurons that can respond differently to different stimuli. Is that my understanding correctly?
Yes.
This is maybe what you're going to get into in the talk, but is it well known how each type is activated and why others don't respond to those same stimuli?
Sometimes, yeah, for some of the different types of neurons, yes, but others no. And yeah, it's like the most basic answer I can give you.
Yeah.
Oh, that's
but
fabulous.
Yeah, it's really cool. I mean, our lab, we didn't really focus on the the biophysics at the level of the mechanotransduction. But I've seen, reading some of these papers of experiments that people do, it is really cool. But for example, like these neurons that I'm talking about today, we don't even know yet what the mechanical transducer is, if that makes sense. So certain touch neurons will express like piezos, like piezo 2, for example. And so that is takes us from a mechanical stimulus to an electrical signal. And we don't know exactly what, so the B4 neurons, they don't express piezo 2. So we don't know what they are, how they're transducing the signal. And yet we've learned so much about them in this paper, but we don't know this, one thing. So yeah.
That's amazing. And I know this is not like the Valentine's Day special, we're a little bit late with this, but like the next question is like, what's the pain pleasure sensory neurons, right? So like, I don't know if I'm overstepping, but isn't there like a connection with pain and pleasure for certain contexts?
Yeah, I mean, I guess like contexts in which certain types of pain are not aversive. I mean, I always like to think about like, I mean, there's so many different situations, but even, right, if this is different, but if you're relieving an itch sensation, right, and you scratch yourself, you're really kind of inducing pain, but it's, to inhibit the itch, but, and it's not aversive. And I'm not an expert on pain and itch and that, but and then I always think, and then deep tissue massage or like stretching after a workout and delayed onset muscle soreness, like that kind of, like it hurts, but it hurts so good, when you're stretching. Mysteries to me that I'm sure there are other experts out there that could even elaborate and speculate some more about how, but whether it's the same pain neurons that are activated in the periphery and at some point in either the spinal cord or the brainstem or the brain where that gets turned into pleasure, I don't know. We don't know at what point. And that's the awesome thing about somatosensation is that it's the whole body, right? From the tip of your toe all the way to your brain, something's happening somewhere, and there's just so many levels to investigate. So these neurons have a lot in common with human C-tactile afferents. But at that point, at this point, we didn't have any evidence of a role for these neurons in any natural behaviors. So we really couldn't define them as social touch neurons until we really implicated them in social touch, especially from a behavioral neuroscience perspective. And so in somatosensory neuroscience, when you want to understand a behavioral role for a group of sensory neurons, like what are they used in natural behaviors, one approach that you can take is called transdermal optogenetics. And so with this approach, we genetically engineer mice to express a blue light sensitive channel rhodopsin. in just the neurons that we're interested in, so in just the before neurons in this case. And so what's cool about transdermal optogenetics is that because our neurons of interest innervate just the superficial layers of the skin, we can just shave the back of the mouse and shine blue light through the skin, and it will activate those terminals. So we don't need to do any surgeries or anything. We just genetically express this channel rhodopsin in the B4 neurons and shine blue light through the skin. And so you can imagine this has been done for identifying, say, pain neurons, where you're going to induce a behavioral response like flinching or licking that helps you identify, okay, those neurons are probably pain sensitive. Or you can imagine, you know, when we optogenetically activate itch neurons, we see scratching behaviors. So then what might we expect to see when we optogenetically activate social touch neurons? So what kind of acute behavioral response would you even see? So we had no idea, but we were really curious and we had the tools to do it. And we reasoned that if we saw something interesting, it could be really informative for what these neurons are doing in natural situations. So we went ahead and expressed channel rhodopsin in the B4 neurons. And then, like I said, we shined blue light through the skin. So on the left here, I'm going to show a control animal, so no channel rhodopsin. You see that the blue light, you know, doesn't really induce any behavior. Maybe the mouse startled a little bit to the presence of the light. But when we express channel rhodopsin in the B4 neurons, what we see is that the mouse begins to step out and lower its back. And then there's this really striking dorsiflexion in the spine. And we just saw this again and again, and we thought it was so fascinating, but we had no idea what it was or if it had any behavioral relevance. So like any good scientist, I started by quantifying
I have a very
it.
basic, maybe practical question here. So
Yeah.
do you kind of like manipulate the light source by yourself, or is there some kind of tracking that's embedded? And it's amazing how fast this happens. I'm assuming these are real-time images.
No, it says 10 times slower than real time. So
Oh, I
this
didn't
is
even
super
see 10
fast.
times slower. It's even
Yeah.
faster than I thought.
Yeah.
Sorry, there's something covering the bottom of the slide on my screen.
Yeah. Oh, yeah. Yeah. So it is manual. So it was, you know, me kind of standing there, shining the light on the back. And yeah, the back is, yeah. So I don't know.
So
yeah.
you took a light, you're controlling like a gene, and you can get a behavior out.
Yeah. Yeah.
That, like, you know, we have central dogma, you know, I'm an engineer, but like DNA, RNA, protein, what, like you're activating something with light and you're changing the behavior of mice. That's insane.
Yeah, it is really cool. And I mean, people do this a lot now in the brain with optogenetics. You know, you can activate one brain region, one cell type, and get a behavior. I think the transdermal optogenetics is just really cool because you also have that additional level of, yeah, you're really in the periphery. You're like manipulating the very first sensation that they have. And then, you know, you're seeing the behavior. But then the question is, because you're so far removed from the motor output, you'd think, you know, there's so much that could be happening, right, in between the skin to the brain to the behavioral output. So I quantified this behavior as the maximal back step from the ceiling of the chamber over the course of like a 20-second simulation. This allowed me to detect differences between these experimental and control animals. And so we had a lot of questions, like I said, but at this point, and so I had mentioned Will Foster was, you know, the one alongside that, you know, I saw this, you know, for the first time. So I just want to mention that again. But so here we had that question of one of the first questions was, well, what do the mice think of this? Is it neutral? Is it pleasant? Is it aversive? You know, what's going on? And so from here, this is where we decided to conduct this condition place preference assay. And like I said, this was led by Melanie Schaffler. And so what she did was she has this 2 chambers, and one chamber is associated with the blue light, which activates the B4 neurons, and the other chamber is associated with green light, which does not activate the B4 neurons, and serves as a control. And she found that over the series of 5 training days, the mice kind of learned to associate that blue light with the activation of the B4 neurons, and found that to be preferable. They spent more time in that blue light chamber compared to the green light control. So this, drives home that point that the activation of these neurons is pleasant. And so at this point, I've shown you that the activation of these neurons is sufficient to cause a dorsiflexion posture and is pleasant. But we still don't know if it has natural relevance. So in the same way that optogenetically activating itch neurons yields scratching, and scratching is a natural response to, say, putting something itchy on the skin, is this posture reflective of a natural response to social touch? So that was the question that we were left with. And so we began to kind of brainstorm, in what scenarios would we ever see a mouse lower its back? And there are 2 main scenarios that came to mind. So the first is what we call this conspecific crawl, where you have this slight dorsiflexion in the back as, female cage mates crawl underneath each other, or cuddling or grooming each other. And then the second is lordosis, which is the female sexually receptive posture in which there's dorsiflexion in the spine in response to the tactile input of the male mount. And so for this paper, we've just shown this lordosis data, but because we're kind of talking about the full picture behind everything, I also wanted to bring up this other angle. And so so we've seen that the neurons are sufficient to induce this dorsiflexion, but our question now is, are they required? Are they necessary for these social behaviors in a natural social setting? So what we're going to do is we're going to ablate the neurons, or get rid of them genetically, and then see if we see deficits in either of these 2 behaviors.
Can I ask a few questions about what you've done up until this point?
Yeah.
So up until this point, how old are these female mice? And then do you expect any differences in this response with age, using younger or even older mice?
Great question. These mice the female mice in the study have all been like young adults. So adults 8 weeks older, sexually mature, probably like 8 to 12 or 14 weeks. And then, but we had that same question, right? And so this wasn't, published in the paper, but we do have evidence that young weanlings, like P21, that they do a very striking dorsiflexion posture. And so that fits together with other evidence. I'm not quite going to get into it, that the optogenetic induced posture itself is not representative of a state of sexual receptivity, because we see it in sexually immature females. We see it across the estrus state when females are not sexually receptive, and we see it in males. So we think that this is a social touch circuit that causes dorsiflexion that is recruited in different social behaviors. That's a kind of a key point that sometimes gets lost.
And then if you actually go back, thank you for the answer. That was super informative. If you go back one slide though to the condition place assay, I'm wondering, is there anything else different about these rooms? And is it always set up where the mouse has to choose one room or the other, or is there a neutral territory that can be kind of placed in the middle for this assay?
Great question. Yeah. So there's a lot of different designs for these boxes. And at one point we had a neutral zone, but then Melanie, I had done a lot of these assays before, and she was kind of saying how sometimes the neutral zone can cause difficulties, and like sometimes it'll just hang out there and you lose data kind of. Yes, differences between the 2 rooms. So the patterns on the walls, the textures on the floors, and then also a neutral scent associated with each side. So that allows them to have multiple cues that help them to associate, the blue light with these other sensory cues. Yeah.
Very cool. Okay, yeah, those were my 2 questions.
Yeah. So at this point, again, what we're going to do is we're going to ablate the neurons and look for deficits in either of these social touch-induced dorsiflexion behaviors. So this is a picture of a dorsal root ganglion, and all these dots are cell bodies, and the B4 neurons are in purple. So in a control, you can see there's a few of them. It's a small population. And the B4 DTA, then those purple neurons are gone, right? So that's what we're looking at, the ablation there. And so I started by looking at this conspecific crawl behavior, and I won't talk too much about it today, but I just wanted to highlight just like I was amazed just how much this optogenetically induced dorsiflexion really resembled this natural social touch induced dorsiflexion, where you can kind of almost map, where the social touch is being applied to the back, and you see these identical postures. But this is it's a novel behavior, and it was challenging to find the right tools to really quantify it, to really assess what was going on. And so through kind of some preliminary studies, I did detect that there was a deficit in this behavior when we ablated these neurons, that the females were less often kind of dorsiflexing in this way. But we tabled it for a future study where we can really use some of the best maybe machine learning methods in social behavior to look at, to really get a deeper look at this really rich data set. But I just wanted to put that there because that was a large part of my project. But lordosis, on the other hand, is a highly quantifiable behavior that's been studied for decades. And so it was just a really nice, accessible measure there. Yeah. So again, what we're going to do is we've ablated the neurons, and we're going to look for deficits in sexual receptivity. So this approach here, lordosis quotient assay, it's pretty straightforward. We're just measuring the total number of receptive female responses over the total number of male mounts. And importantly, I'll just mention that one of the key drivers of female sexual receptivity in mice is the state of the estrous cycle. So the sexual hormones that cycle across 4 days. And so it was important that we control for that by overreactomizing the females and then adding back the estradiol and progesterone, so that all of the females were in that same level of hormonal receptivity. And so this is what I had mentioned before, where Will had come in with this previous knowledge of how to do these ovariectomy surgeries and, taught me and Bella, and so that we were able to do these surgeries. And so for this assay, so what we saw, so on the y-axis here is lordosis quotient. So the higher the bar, the higher the sexual receptivity, the percentage of receptive responses. And I'll just say, so to measure, we deemed a mouse to be receptive in if all 4 limbs were on the ground and there were no attempts to escape. So what we saw is that in wild type controls, on that the first pairing with a male, there's kind of this mediocre level receptivity that then increases upon subsequent sexual encounters. And this has been published before, that mice will increase their sexual receptivity with increased encounters with the male. But when we ablated the B4 neurons, what we saw was that on the first trial, we had very similar kind of mediocre level receptivity to the controls. But then on the second trial, instead of increasing receptivity, we see the receptivity plummet. And that stays low for the third trial as well. And so this can also just be seen really dramatically looking at changes in individual animals with this upward trend in the wild type and the downward trend in the B4 ablated. And then we also measured just the average duration that they maintain a receptive posture. And we see that by the third trial, the B4 ablated females have a shorter duration of their receptive posture.
So I'm wondering, one of the questions that I've had, like when looking at the paper, is just like context of touch. And so I'm wondering if you think like your very first graph that you showed us in the wild type C57, that's like a level of context where you said that mice, once they've been conditioned to mounting by the male, they will often go back into this position. So I'm wondering if that's kind of an example of context where they're being conditioned to expect this response.
Yeah, definitely. Yeah, I think, a large part of this seems to be kind of, like a learning, like almost like a learned behavior. They're associating their experience with something positive, and then they choose to engage in subsequent trials. And that's exactly what seems to be lost when we ablate the before neurons, because on that first trial, it seems like they're actually pretty similar, right?
Yeah.
But it's not until that second trial where we see that they're saying, the wild type animals are choosing to engage, and then the before ablated are choosing to actually, we see combative behavior. So they're really avoiding the mounts altogether.
And then I'm also wondering, in the animals that you've ablated these neurons, do they have any other like social defects when it comes to interactions with the mother? Because these, or just any type of thing, as any type of other defects that you notice.
Yeah, that's a great question. So in Melanie's work, she found that there was no difference in kind of a basic or a kind of traditional social place preference assay, if that makes sense. So they won't choose, they don't have any deficits in social interest among adults. And I also saw in my that female social interaction assay that there were no real changes to how much they contacted their cage mates. So they were just as likely to contact their cage mates. You mentioned maternal behaviors. During my time in the lab, didn't look at maternal behaviors, but it was always something that we wanted to do in the future. And I believe there are people in the lab currently working on that.
But there were just like no overt kind of, so that, I mean, that's a good thing. There was nothing like overt that you just kind of noticed, like, yeah, okay.
Exactly. Yeah, right. Nothing was overtly different. We assessed, Melanie assessed the motor behaviors of these mice. And they were, motor behavior was fine, but with our assays, they're normal. Their social behavior seems normal, which is something that's just really interesting about this, is that it seems very specific to social touch and touch dependent social behavior. So it's not just, we're not creating like a new autism model or anything like that. Their social interest is there. It's just they have a deficit in social touch, and that really impairs social touch behaviors and the way that they choose to engage in those social touch behaviors.
And maybe you'll get into the implications of this, because this data is incredibly striking. You have this like kind of one gene control of sexual behavior. And I was kind of thinking of like other animal models of autism where they'll literally just walk around the edges of the cage and won't explore like the middle area. But my question, that was kind of an aside, are there any other environmental controls of this gene expression? So like maybe diet, you talked about the hormonal aspect as well, but can you stratify kind of what the consequences or what other regulatory elements are to control this gene? Because it's very clear, this is on and off, this is very binary, and you see this behavior, but can you kind of contextualize it into, other environmental impacts on this behavior?
Okay, so I'll start by clarifying one thing. So this gene itself, we're just using to like get a handle on these neurons. So we're ablating the cells, and we actually know very little about this gene and very little about this protein and what it's doing. All we know is that it's a G protein coupled receptor that's expressed just in these neurons, and we don't know what it's important for. There's previous literature that knocking out this gene doesn't impair anything drastically. I don't think it was ever assessed in sexual behavior. But so you were talking about environmental changes that could affect this behavior. Okay, I can talk like other things that modulate sexual behavior and sexual receptivity are like olfactory cues. You said hormonal, olfactory, and then touch are kind of like three key pillars to sexual receptivity in mice, I could say. We had done kind of a fun, I don't know if that noise is coming through, it's my cat eating, but we had done an experiment where we tried to see whether that the opto-induced posture could be modulated by like a male scent. So we took male bedding and, you know, see it, and we didn't see any striking changes. But yeah, those are good questions. And I think it's also important to note that, you know, these females, like, they really lose their sexual receptivity, but there's always a remainder that, you know, that stay, right? And I think that's, you know, this is such a critical behavior, right? Sexual behavior for the survival of a species. And so, of course, there are probably other neurons that are compensating and that are playing a role in this. And so, yeah, so I think that's just, yeah, an important thing to note as well.
I actually had a question that builds exactly on what you just said. It's always really striking, I think, to see something go to zero. Like, you have a population of these mice that are their lordosis quotients going to zero. But as you mentioned, maybe there's some compensatory neurons. Is there also some limitations to the ablation technique where maybe the efficiency is not perfect always, or I guess, is it for sure some kind of compensatory response?
Yeah, we did see a pretty good ablation. I'm forgetting the number of this particular, like the percent, but it was pretty close to 100%. But I would probably just guess, just knowing the number of different types of sensory neurons that innervate the skin, and the fact that I'm not showing the data here, but we do still see that these V4-DTA animals are able to go into the posture, especially, you know, looking on this first trial. If we assess the quality of the posture, and they actually can do a pretty good lordosis posture on that first trial. So it's not that they're unable to sense the pressure on the back and they're unable to make that posture. It's more so they seem to be choosing not to on the subsequent trials. So that's what really pushed us towards the more affective component of touch. And that actually kind of leads me into the next slide. So thank you for the transition. But yeah, so the neurons don't seem critical for the generation of the posture itself, especially looking at that first trial. So then we were asking, well, okay, if these neurons are important for the positive reinforcement of sexual receptivity, are they then directly conveying a pleasant or rewarding sensation that causes the females to choose to engage on subsequent trials? And so to test that, we combined our previous technique, our transdermal optogenetics, with fiber photometry. And so here, what we did, so it's the same technique as before, genetically expressing that channelrhodopsin in the B4 neurons in the skin, shining blue light on the skin to activate them. But now what we're doing is we're also recording from the brain. So we're recording specifically dopamine release in the brain region called nucleus accumbens. And so the release of this neurotransmitter into this brain region is commonly associated with reward behaviors. So this is the approach that we decided to take. And we then began to measure dopamine release when we activated these neurons. And so I'll just say this was really novel because no one had ever really paired like a specific population of dorsal root ganglion neurons with a specific effects in the brain. So this is a really cool approach. That's just showing. So the approach here, we're expressing this protein that when it binds to dopamine at the synapse, it will fluoresce brighter. And so we can then use the light as a proxy for dopamine release. So when we see a lot of green light in the nucleus accumbens, then what we're seeing is dopamine release. And so we know that that during the sexual encounter, there's tactile input to both the mount, both the back during the mount, as well as to the anogenital region. So we measured dopamine release in the nucleus accumbens when we activated the neurons in both locations. So first, the back stimulation. And so you can see here that when we shine the blue light, activating the before lineage neurons to the back, we see dopamine release right at that kind of light onset, but we don't see it with the blue light or the green light as a control. And then we see the same thing when we shine the blue light to the anogenital region. And so you'll remember that the B4 neurons innervate just about all the hairy skin that we've ever looked at. So we've also found that they do innervate the hairy skin in the anogenital region. And so we can see that there is dopamine release with that blue light, but not with the green light. And so this was a really exciting finding because we're showing that, yeah, even in a socially isolated mouse, we're able to activate this skin to brain circuit to activate the sensation of pleasantness or reward, independent of a social interaction, which is really exciting. Okay, and I have one more data slide. So this demonstrates that the B4 neurons are sufficient to engage that circuit and cause dopamine release. The final question was, are the B4 neurons required for dopamine release during the sexual encounter? And so we took this fiber photometry technique and we used it in those B4 ablated animals, the B4 DTA animals. And what we saw is that in the wild type controls, we see this little dopamine release at the mount onset, and then that goes away when we ablate the B4 neurons. So suggesting that these neurons are required for dopamine release during the male mount. And so we can speculate that that could be a reason that they're choosing to not engage in this sexual encounter on subsequent trials. Oh yeah, yep.
And so I guess the opposite of that, without these neurons, you're not getting any type of like pain sensation. They're just not getting the pleasure sensation.
Yeah, that's a good question. We're not assessing pain, but this really, I mean, we have shown that there is the lack of the pleasant sensation. For some of the revisions experiments, a team at Columbia, led by Bella, she did these experiments assessing just like pain, basically doing a pain assay in the B4 DTA animals, and saw that their kind of pain stimulus, their pain response overall was pretty normal. Yeah.
And so I'm wondering, even if in other studies, like since we asked that question, there's many different types of neurons that are able to detect different types of stimuli. So in the absence of one type of neuron, does that stimuli just not get perceived normally, like how you're showing, where like they just don't have that dopamine release? Or have there been other examples where another type of neuron will then compensate for the lack of that other neuron, and you'll have some type of stimulus there?
There's definitely evidence where when you get rid of one group of neurons, there are compensatory mechanisms, or even you could imagine changes in central circuitry, where different populations of sensory neurons have different roles. There's a lot that could be going on.
And then I kind of had maybe a weird question again. So you talked about this like dopamine release, and you kind of see this like accumulation over other trials. So does that kind of, I'm not a neuroscientist, but correspond to like addiction? Like you have more and more dopamine under like a pleasurable scenario, and then you see in the opposite trend, there's like less dopamine release. So there's no more of this activation. So can you kind of like contextualize what we think about dopamine and pleasure and, along those lines?
Yeah. Dopamine release is associated with addiction, but it doesn't have to be, not all of it, right? Like normal healthy dopamine release is good. What do I want to say? Let's see. I'm definitely not an addiction expert by any means. So
But you see like this varying threshold, right, in terms of the degree of dopamine. So does it require more or less with subsequent behavior? Like, can you like stratify based upon your results?
Yeah. I mean, yeah, we only did up to 3 trials. But yeah, yeah, I don't know. I wouldn't expect, yeah. It seems kind of like with addiction, it's kind of if it's a dopamine overload, right? And then you're requiring it to maintain that same level, then other behaviors that would normally be rewarding no longer are sufficiently rewarding.
Right.
And then you also have levels of withdrawal. But I think here we're just looking at, yeah, like healthy levels of dopamine release that yeah, that it's we're not looking at how it affects other behaviors too. Yeah. I don't know if that
No, that's fine. Thank you.
It's yeah, and this project spans like everything from the biophysics of somatosensory transduction to behavior on every level. So yeah, it's awesome. Okay, so I'll just kind of recap what I've gone through today, and then get into some of the behind the scenes stuff. So I showed that optogenetic activation of these neurons is preferable and evokes a dorsiflexion posture, which now represents the first stereotyped behavioral response to the activation of social touch neurons. So now that they're implicated in social touch, we can say that acute behavioral response is kind of akin to maybe like itching is or scratching is to itch neurons, right? So it's kind of cool in that way. I didn't talk about this today and it wasn't in the paper, but it seems like the neurons are also mediating aspects of female-female social postures. And then I showed that the neurons are mediating rewarding sexual touch and are required for female sexual receptivity and for dopamine release during the male mount. And lastly, the activation of these neurons in either the back or anogenital skin is sufficient to cause dopamine release, even in a socially isolated mouse. And so we can take a step back to our original question: how does the skin detect socially relevant tactile cues? And so I, haven't come close to answering this in entirety, but I can say a few things of what this paper really, what progress it has made towards that question. And so one, it shows that it can be conveyed through specialized sensory DRG neurons. And so we know that they have similar anatomy and some physiology to human C tactile afferents. These B4 neurons do. And so in my work, we showed that these neurons also are implicated in social touch. And so that there are these specialized sensory dorsal ganglion neurons that are similar from mouse to human, and that both convey social touch, are important for social touch behaviors. And secondly, this seems to be happening through a mechanism that doesn't require social context. So the fact that we can activate these neurons in a socially isolated mouse and reap some of the benefits of social touch, dopamine release, is really cool. And I always like to kind of compare this to the effect we see with weighted blankets. So we know that when you wear a weighted blanket, you can get anxiety anxiety reduction. And so that is thought to be kind of mimicking a hug, right? So you're reaping some of the benefits of a hug just by activating the sensory neurons, right? With the weighted blanket, all you're doing is activating the periphery, and yet you are able to reap some of the mental benefits of a hug. Of course, it's not the whole experience, right? Like hugging a loved one is, way better than putting on a blanket. But the fact that we're able to engage part of that circuitry is, I think, really important to know. And so through this work, we kind of showed that maybe stroking sensitive neurons, we don't think these are detecting pressure, may be able to have kind of a similar effect where you can activate them in social isolation and reap some of the benefits of that activation that you would normally see in a social context. Yeah, and so that's the core, the very, very core of the paper. But if you look at the paper, you'll see that there's a ton more experiments and so much other work that so many people did. And in addition to that, there are experiments that didn't even make it into the paper. And so now I'll just talk a little bit about some of the alternative approaches that I tried, and just things that didn't come into the paper. And so one of these things is just looking at oxytocin, right? So oxytocin has long been known as this social hormone, and despite some recent work, we still know that oxytocin is critical for social behaviors. And so for a long time, rather than looking at dopamine, I was looking at oxytocin. And so I created, along with Bella, a quadruple transgenic in order to observe dopamine, observe oxytocin neuron activity during the activation of these neurons. As a neuroscientist, this is out of my field, and I learned how to conduct ELISAs. So that's taking blood samples and looking at oxytocin levels in the blood, just studying and understanding hypothalamic circuitry, and conducting fluorescent in situ hybridizations for oxytocin expression in the brain. So these were all things that took up, months of my time. But, you wouldn't even guess that I had done oxytocin, right, at all, if you read this paper. And then I already mentioned this, but a large part of this project was looking at female-female social interactions. And so because we felt like this could be optimized a little bit more and quantify it a little more rigorously, we decided to table it for another study. So these are just some of the things that, alternative approaches and things that didn't come into the paper. Curious.
So are there groups that are sort of working on, I think you mentioned machine learning, but like tracking mouse movement in these, and like, I imagine if you could do that with some software or something, you could get a lot of interesting data there. So are there groups that specialize in sort of mouse tracking and, quantifying these behaviors?
Yes, absolutely. And I'm not an expert. I'm not an expert in any of, I feel like, any of these things. So social behaviors, there's a lot of new machine learning algorithms to do markerless tracking, as well as tracking with markers. And there are still challenges in identifying keeping track of unique mice, as well as for example, this posture, you really have to have a certain angle for the camera. And so I think maybe now they have from multiple different angles. But I know, Kay Ty's group has a method that they've been using and has been widely used. Victoria Brera as well studies social behaviors, and there's a lot of other methods out there for quantifying these kinds of behaviors. And probably, if I had stayed on a few more years and really invested in this, it totally could have been part of the work. But we'll leave it for the next the next person. And all of Ishmael's postdoctoral work and a lot of his current lab is really using these machine learning approaches for quantifying pain behaviors. And that's actually, I didn't even get to mention this, but that was really critical for identifying this posture in the first place. You'll see that we put these mice in these chambers where we can do high-speed videography and get these really high resolution, high temporal and spatial resolution of these behaviors. And he did that so that he could really assess the details of the withdrawal response. And so because we had started out from that, with that approach, we were able to really get a good look at this posture induced by the optogenetic activation of the neurons. So Yeah, using machine learning methods and high-speed videography for quantifying mouse behavior is a huge field in itself. Yeah, I think, you know, in the introduction of the paper, we cite like all these different papers or all these different approaches. So it's becoming really huge. So I hope soon this is a simple problem. Yeah, and for somebody somewhere, it probably is a simple problem. Yeah. So another thing that's kind of a black box is the submission and review process. So I'll just kind of give a little bit of an overview of it. So you know, if you've never submitted a paper before, it takes time, right? From the completion of the experiments to the time that that paper is out there. So I had a hard deadline because the lab had to be in boxes on July 1st, 2021. And so at that date, I was all, you know, the initial submission, the experiments were complete and the lab leads for Columbia. That summer then, I was doing the data analysis and writing. That fall, we submitted it to Nature first, and then we get this desk rejection, right? So before it gets submitted to peer review, we get this desk rejection after a few weeks. And then you submit it to another journal and then desk reject. And then we sent it to Cell, and then we got the notification that it was sent out for peer review, which is like a big in, because that means that at least the editors are interested in it, right? And so that was exciting. At the end of December of that year, we get our revisions, like the revisions back. So they ask us to do things or ask us to revise things. So some of these are kind of straightforward, right? Changes to the writing or statistical explanations, different citations to include. All of the reviewers wanted more of the male data. So we had previously, you know, looked a little bit at how the males were responding, you know, to the optostimulus, but we hadn't really done a rigorous sex differences comparison. Instead, we decided to kind of focus on the female story.
And so, yeah, I'm wondering what that initial decision, like where'd that come from to only focus on females? Is that like, do you guys just normally only work with females in the lab, or? Yeah, I'm wondering.
Yeah, it's a great question. So I think when we saw the posture initially, and we were thinking of those two behaviors, you know, what we call the conspecific crawl and the lordosis. So we knew that lordosis was naturally only a female behavior. And so we kind of decided to focus for the lordosis experiments, you know, it all had to be females. And so we were kind of making sure that for all the histology and everything that we did was also in females to keep that consistent. And then for the cage mate social interactions, we had, because we were sticking with females for that side of things, we started out with females there. And we also kind of speculated that maybe in male mice, there's a really strong dominance hierarchy. And we thought maybe that social touch to the back would not always be so pleasant. Maybe it's part of that, you know, the dominance hierarchy and like being a more dominant or a submissive male. So we kind of speculated that it might play a different role. Doesn't mean that it's not important and it's equally interesting. But for this paper, we decided to kind of focus on the females. That's how that direction started. But then you can see in the paper, we conduct the conditioned place preference in the males, and then we also quantify the opto behavior in the males, and we look at like expression of this protein or expression, the number of the cells in the DRG as well. Yeah, so I guess to recap, they do the posture, the males do the posture. It doesn't seem like there's a strong place preference for it though. It seems to be neutral. And then the numbers of these neurons seem to be the same in males and females. Yeah. And then, a few other things that we did. So we increased the animals for photometry. I was kind of limited by the timeline, I only conducted, kind of a pilot for some of these photometry experiments at the time. And then two of the other bigger things that we did, so looking at individual dopamine neurons in the ventral tegmental area, the VTA, which project to nucleus accumbens. So looking at those neurons to see if they responded differently to different types of social touch. And so that was led by Akira Fushiki, did the Inscopics experiments. And then also the experimental distinction of the B4 lineage neurons from the B4 adult population, which I didn't even get into today. This study was on the B4 lineage neurons, the ones that express B4 in development. And then we can also distinguish that from the neurons in adulthood that maintain expression of B4 neurons. So Bella led this effort to use a viral approach to more selectively manipulate just the B4 adult population. And that's all laid out in the paper, and I could go into that, but it's another whole story. So, and then 10 months later, we resubmit the paper. And at this point, it's kind of like, okay, this is the first resubmission. I've heard that sometimes it takes a lot of, trials, and we really tried to do everything that they asked. And so, but I was really grateful that just within a few months it was accepted, because at this point I had been done with my PhD for over 6 months. I was in my postdoc. I hadn't been in the lab for over a year. So I was really grateful that we were accepted at that point. Yeah. Yeah. And then lots of formatting. That was something I was like, wow,
yeah,
I have to do this. I'm paying for this, and I have to do it.
Yeah, exactly. Yeah, you're like, what are the editors doing? Maybe, but yeah, no. Thank you for going through this process. This is super insightful. Yeah, it seems like you guys attempted and really did well, addressed a lot of the reviewer comments, and so yeah, you guys had that quick turnaround time, which is really promising to see.
Yeah, we were grateful for the editors and the review. Everybody was reasonable. There wasn't, no one was unreasonable. So yeah. And then the last thing that I think I have here is the kind of timeline of, I just kind of showed the timeline of the submission and review process, but I can talk about the timeline of the project itself and kind of its origins and everything. Definitely, this is more so my perspective as a PhD student. You could talk to any of the other authors and they have their own story. But I hope it's helpful for any other PhD students out there who are wondering, so is it like a linear, is it a linear path? Like, how does this work? No, it's not. But yeah, so in the fall of 2018, I was a second year PhD student, and I rotated in the Abu Sabor lab at that point. And that was when, alongside Will, we saw this dorsiflexion posture to the optogenetic activation of the neurons in the back. And I thought, well, this is really cool, and everybody in the lab is phenomenal, and I'd love to work with them. Ishmael is a great mentor. I'm going to join the lab. So I joined the lab. And then the whole year of 2019, from January to December, I kind of called that the building and troubleshooting year. So the lab was brand new. The lab opened in the summer of 2018. And so at the beginning of 2019, it was a lot of, hey, let's set up the confocal, like let's get this going. And I set up like RNA scope in situ hybridizations in the lab. So that was really cool. And once those were working, I could characterize the B4 lineage neurons with in situ hybridizations. And I didn't even show that data, but you can see it in the paper, but staining these neurons and looking at expression of different mRNAs in these neurons to really get a feel for what are these neurons that we're activating. That summer, and again, another piece of data I didn't talk about today, Bella and I found that the posture is not dependent on ovarian hormones. So we had thought, okay, well, maybe this is lordosis posture. It should be modulated by ovarian hormones. And it wasn't. So I was like, oh man, like that's a big hit. At this time, Melanie was trying everything to ablate the B4 neurons. And she finally got it to work with the DTA genetic strategy. And so that was a struggle. And then we also, this is something you can sometimes see in mouse work, that mice will often cannibalize their young. And so breeding can be a really big challenge. And so at this point, there was this 10-month period where I didn't have any new experimental animals. So like no new pups. So that was really hard. And I think a lot of it, like this is my first time working with mice, and I didn't know how to manage a mouse colony and kind of just have an idea of like, how many breeding cages do I need to set up? And how concerning is it when like 2 litters, back to back are cannibalized? Like, what are things that I can do to address this? And so after that, I was like, okay, like now I know like breeding is a priority. Like you have to be real. It's a real challenge. So that was kind of the struggles of 2019. And then the pandemic hit. So this is, I call this season Quiet Can Be Good. So for 3 months, kind of at Penn and our labs, we kind of shut down pretty much, kind of 1 or 2 people allowed in. So I actually went home to Massachusetts and was with my parents, and I wrote my NRSA and submitted that in April. So it was good because I had time to really think and finish writing my first like NIH grant, which was awesome. And then during this time, we speculate maybe because the facility was just so quiet and peaceful that we finally started getting these experimental animals. So that lockdown in my situation was actually kind of beneficial in those 2 ways. So in summer of 2020, I come back to lab and I'm like fully energized. I had my 3 months to kind of sit back and think, and now I'm like ready to do all these experiments that I want to do. And at this point, as I had mentioned, I was really looking at oxytocin. And so I was looking at oxytocin expression in the brain. And with the in situ that I was doing, I did that experiment and I had, and I didn't see any differences with the method I was using. And so at this point, it's been 2 years since I had this initial finding of a dorsiflexion with 0 experimental evidence of any role of these neurons in any social behavior. So at this point, Ishmael and I were like, okay, for the sake of me finishing on time, like it's been 2 years without any positive, data, like we should probably drop this project, except for the fact that I did finally have pups that, where we ablated these neurons. So, okay, we can do that. The final experiments, right? The last straw, which was to do the deficits in social behaviors. So at this point, we do that final experiment, or what we thought was the last thing. So we ablate the neurons at last, and we see deficits in the female social posture, and then I see deficits in sexual receptivity. And around this time, Melanie also sees that there's a place preference for the activation of these neurons, and suddenly, we're back in the game, and we're like, okay, like I have a thesis project, like. I know what I can do. And then we find out that the lab is moving to Columbia in less than a year.
Oh no. Okay. Well, before that, I'll say that it's so like just nice to see that actually the paper, the project that became this really big impactful paper, you were thinking about dropping, like, in summer 2020. And I think that, yeah, like you said, it's really helpful for people who are entering PhD programs or are currently in PhD programs to kind of see that it's not linear. Yeah, at some point,
Right.
you may not necessarily want to give up, but maybe the project kind of gives up on you a little bit, and it takes some activation energy or just a different way of thinking of things to get things back going. So yeah, that's super inspiring and great.
Every PhD student needs to see this. I think like science is like resiliency, and you've really shown it here. And like, I want my grad students to see this as well. Like, it is science, and you can't control it sometimes, but you have to have that like tenacity. And this is incredible. Like, this is super inspirational. I don't know.
Yeah, no, it's a story. And so, I definitely want that to be, like for people to see that science is not linear, and, things can be really challenging, but you really never know what's around the corner. So then in the winter of 2020-21, this is kind of like the part of the pandemic where things were, people still weren't really vaccinated yet, and we were still coming into lab in shifts. And this was a really lonely time in lab. So, most of the time I wasn't seeing people, right? I'd be in there like doing my in situs, and I'm just like, it's so quiet, like there's no one here. And I was also, this is where I was, technique building again, right? I'm trying to set up fiber photometry in a lab that doesn't study the brain. And, we're all the peripheral nervous system. And so this is a really big challenge as well. And then I was also doing the oxytocin ELISAs, which was not at all in our fields either. And this is where I'll also mention that the rest of life doesn't stop for a PhD, right? So during your PhD, a lot of times you're in your 20s or whatever period of life that you're in, like a lot of changes can happen. And so that's different for everybody. But for me, that winter, I got engaged, which meant that the next 8 months were wedding planning.
Which is very stressful, very stressful time.
Yeah, so that's also really time consuming too. But it also, it keeps things in perspective, right? Like your whole life can't be your PhD because your PhD doesn't love you back, right? Like you have to have, you have to have your life that keeps you going. Yeah. And so around this springtime, people start coming to lab again, and there's this kind of revival. Like I think I have one in-person meeting with Ishmael in his office, and I'm like, wow, like it's been a year, like how are you? And then I call June 2021 the mad race to finish, because we had this hard deadline of, July 1st, we're in boxes, right? So at this point, I had kind of just gotten photometry to work consistently, and I had the mice. And so I was collecting these data sets as well as I could before the lab leaves. And I also was writing a review paper in that time. So I just remember it as like a crazy month. And then this is just the sad thing. Then all my friends leave again for Columbia right as they came back. But no, it was good. New beginnings for the lab. Yeah. And so that summer, I was analyzing the data and writing up the manuscript. And then in the fall, we submitted the paper. And I was also granted permission to defend at that time, which was exciting. And then in October, I had my wedding and honeymoon, and that was just, good times in personal life. And then I moved to Baltimore. So my husband was in Baltimore. My lab was in New York City. What was I doing in Philadelphia? So, so then I moved to Baltimore and began interviewing for postdocs, while my paper was under review. And then we get the revisions back in December, which were overall positive, right? Like, seems like we had a chance of getting this paper accepted. And so January to April was really kind of peak revisions time. And I was this East Coast nomad, like taking the train from Baltimore to Philly to New York to do surgeries and photometry experiments primarily. And then I was also writing my thesis. And then again, wouldn't have been possible without the whole team doing these revisions experiments. So kind of managing this team at Columbia and Rutgers and Penn to get everything done. And then, I get my PhD in April, I defend. And then, the last summer, then the kind of revisions were continuing at Columbia, things were getting wrapped up, and I start my postdoc. And then, as I said, kind of we submitted in the fall, and it was accepted soon after. Yeah. So that's the story. I'm happy to answer any more questions.
Thank you so much for sharing that with us, especially the personal aspect of it. I think, just as you said, the rest of your life doesn't stop for a PhD, and so
right,
either does the world. So you kind of navigated and overcome so many different things during your PhD. So it's just really great to see this nice, complete story and paper come out.
I have a couple questions actually. The first one may be related to some kind of future applications. So are there mouse models of autism that could be used to kind of study perhaps deficiencies in this specific response, or are there other types of models that you're interested, or maybe not you specifically, but the lab is interested in using for this to kind of build on this work?
Yeah, I don't think our lab, actually, I mean, I don't know, but I don't think we're using, genetic models of autism at this point, because, yeah, what we saw in these experiments anyway, we didn't see deficits in social behavior. So we were kind of, in social interest behaviors. So we didn't have that much of a focus on autism spectrum disorder, but yes, there are lots of different genetic models of autism. Melanie wrote, and I co-authored a review paper in 2019 about different genetic models of autism and the role of the peripheral nervous system in autism. A lot of that work was conducted by Lauren Orefice at Harvard. So kind of really just pointing towards the peripheral nervous system and how you can have these selective mutations in the peripheral nervous system that do cause autism-like behaviors, or you're seeing effects in the peripheral nervous system in a traditionally used genetic model of autism. There's so many different autism-related genes that and you can recapitulate some of the behavioral aspects of it by putting those mutations in mice. But yeah, I'm definitely not a autism spectrum disorder expert. But yeah, I mean, the interesting thing though, to try to tie my project to ASD, I guess, we think about autism, we think about how the social touch is not rewarding. And so I guess the question is, in our pathway, we have a pathway for rewarding social touch, and we have, no idea how that pathway might be disturbed in ASD, or whether mouse models of ASD, as you're getting at, have changes to this pathway. So I guess maybe what you're getting at is, if we take a ASD genetic model, can we see if we activate these B4 neurons, would we see a different effect? Because we don't know whether that change is happening in the periphery, in the central nervous system, or both, probably both. Yeah,
that's really interesting. And are there, so the three of us have interest and expertise in kind of cell-based models, and you mentioned there's some kind of unknowns, especially related to kind of mechanotransduction. So have you, or do you work with anyone who has sort of in vitro models? Obviously with such a behavioral study, there's some difficulties there. But are there good models to kind of connect some of those dots, or are there some being developed maybe?
Yeah, like you said, like in vitro work in neuroscience is always tricky. Neurons do different things in culture. And so to tie that to behavior is always treated with a level of skepticism. But, we do have, so there's in kind of figure 1 or 2, like looking at the mechanical sensitivity of these neurons. So just taking one neuron in vitro and poking it, and we see that these B4 neurons, just to confirm that they are mechanically sensitive. That was a question that we got. Yeah. I think with mechanotransduction, it's actually pretty difficult to do things in vitro because you really need the whole skin. I didn't talk that much about the skin, but how you need like all of the cell types in the skin that surrounds the neuron, the terminal, really affects the mechanotransduction itself. So people will do what's called a skin nerve prep. So it's a different kind of in vitro prep, where you're just taking a segment of the skin and you still have the whole nerve intact up to the DRG, the cell body. And it's a very technically challenging experiment. But people have tried it for these neurons. I don't think anyone has successfully done that skin nerve prep for this subset of neurons. Yeah.
Well, yeah, so with that, we would just want to thank you again for coming on to the paper trail. We're really excited to have you talk about your work and kind of share the story behind it.